In 1952, inside a dimly lit laboratory at King's College London, a young scientist was doing something that almost no one else in the world could do with the same level of precision. She was photographing the invisible. Using a technique called X-ray crystallography, Rosalind Franklin had spent months carefully directing beams of radiation at tiny crystallized samples of DNA, waiting over 100 hours for a single exposure to develop, and then analyzing the resulting patterns with a mathematical rigor that her colleagues could barely match. Most people working on the DNA problem at the time were building physical models out of metal rods and cardboard cutouts, essentially guessing at the shape of the molecule and then checking whether it made chemical sense. Franklin wasn't guessing at anything. She was measuring, calculating, and documenting with the kind of obsessive exactness that produces real, irrefutable answers. In May of that year, she captured an image that would later be called Photograph 51, and the moment she developed it, she was looking at one of the most significant pieces of scientific data ever recorded in human history, a crisp, unmistakable X-shaped diffraction pattern that told anyone trained in crystallography exactly what they needed to know about the structure of DNA. The molecule was a double helix, two strands wound around each other in a spiral, and the measurements encoded in that single photograph were precise enough to calculate the exact dimensions and geometry of that structure down to fractions of a nanometer. Franklin knew what she had, and she was in the process of writing it up carefully, the way a scientist of her caliber always would, verifying every measurement, cross-referencing every calculation, making absolutely certain before publishing a single word. She was weeks, perhaps days away from announcing one of the greatest discoveries in the history of biology. And then the photograph was taken from her. Not violently, not dramatically, but quietly, without her knowledge, by a colleague who handed it to two men in Cambridge who immediately understood exactly what it meant and used it to race her to publication. Rosalind Elsie Franklin was born in London in 1920 into a prominent Anglo-Jewish family where intellectual ambition was common, but scientific careers for women were considered unusual at best and inappropriate at worst. Her father, a banker with strong opinions about the proper roles of men and women, initially resisted the idea of sending her to university at all, believing that education of that depth was wasted on a daughter who would eventually marry and run a household. Franklin had decided she wanted to be a scientist at the age of 15 and simply never entertained any other possibility, which tells you something essential about the kind of person she was, someone who moved through the world with a clarity of purpose that most people spend entire lifetimes searching for and never find. She studied physical chemistry at Cambridge, graduated with distinction during the Second World War, and then spent several formative years in Paris mastering X-ray crystallography under some of the finest practitioners in Europe. The technique itself was extraordinarily demanding. It required not only sophisticated equipment and enormous patience, but a deep mathematical fluency that allowed you to interpret the complex diffraction patterns produced when X-ray beams scatter off crystalline materials and reconstruct from those abstract patterns the three-dimensional structure of whatever molecule you were studying. Franklin became so accomplished at this that by the time she returned to England in 1951, she was widely regarded as one of the best crystallographers on the continent, and King's College London recruited her specifically to apply that expertise to one of the most important unsolved problems in all of science: the structure of deoxyribonucleic acid, the molecule that carries the genetic instructions for all living organisms.
What made the DNA problem so electrifying in the early 1950s was that nearly everyone in biology understood that whoever solved it would be unlocking something fundamental about how life itself works, how traits are inherited, how cells replicate, how genetic information is stored and transmitted across generations. Several groups around the world were in active competition, including a team at King's College, where Franklin was working, and a pair of researchers at the Cavendish Laboratory in Cambridge named Francis Crick and James Watson, who were brilliant, ambitious, and working in a style almost diametrically opposite to Franklin's. Where she gathered data methodically and refused to speculate beyond what her measurements could support, Crick and Watson worked by intuition and model building, constructing physical representations of possible molecular structures and then testing whether they held together chemically. It was an approach that had a kind of swaggering creativity to it, but it also had a serious weakness. Without reliable experimental data to constrain the models, you could spend years building beautiful structures that turned out to be completely wrong. And in fact, Crick and Watson had already embarrassed themselves once by unveiling a model so obviously flawed that Franklin herself had to point out the errors at a presentation they gave in 1951. What they needed, and what they knew they needed, was data. Specifically the kind of precise, quantitative data that only someone with Franklin's skills and equipment could produce. And in early 1953, they got it, through two separate acts of scientific betrayal that would shape the history of biology for the next 70 decades. The first involved Maurice Wilkins, a physicist at King's College, who was nominally working on the same DNA project as Franklin, but had a deeply dysfunctional relationship with her from the beginning. There was a misunderstanding at the outset of her appointment. Wilkins had assumed she was being hired as his assistant, while Franklin understood herself to be leading an independent line of research, and the resulting tension between them never resolved. Wilkins had access to Franklin's work because they were in the same department, and in January 1953 he showed photograph 51 to James Watson during a visit to Cambridge without asking Franklin's permission and apparently without seeing anything problematic about sharing a colleague's unpublished data with a competing research group. Watson's own account of the moment, written years later, describes how his pulse quickened the instant he saw the image, because the pattern it displayed was unmistakably that of a helical molecule, which immediately told him that the triple helix model he and Crick had been working with was wrong, and that they needed to rebuild their thinking around a double helix. The second act was in some ways even more troubling because it involved an institutional channel rather than a personal indiscretion. Max Perutz, who headed the Medical Research Council unit at Cambridge, where Crick and Watson worked, shared with them a detailed progress report that Franklin had submitted to the MRC. A document containing her precise measurements of DNA's unit cell dimensions, the water content of the molecule, and a range of other quantitative parameters that she had spent months deriving from her crystallographic data. This report was prepared for internal administrative purposes and was never intended to be circulated to competing researchers. Franklin had no idea it had been shared. Armed with her photograph and her measurements, Crick and Watson went back to their model and within weeks had constructed the famous double helix. On April 25, 1953, Nature published their paper, Molecular Structure of Nucleic Acids, a structure for deoxyribonucleic acid, along with two companion papers in the same issue, one by Wilkins and one by Franklin and her student Raymond Gosling. The arrangement of the papers made it look as though Franklin's work was merely experimental confirmation of the theoretical model that Crick and Watson had independently derived, when in reality the causal relationship ran precisely in the opposite direction. Her data had made their model possible. The acknowledgment in the Crick and Watson paper was a single vague sentence noting that they had been stimulated by a knowledge of the general nature of Franklin's unpublished results, which was an almost comically inadequate description of how deeply her work had shaped theirs.
Franklin left King's College and moved to Birkbeck,
where she determined the structure of the tobacco mosaic virus and began studying polio, more groundbreaking work, done quietly and without recognition. In 1956, she was diagnosed with ovarian cancer at 36. She kept working through treatment, attending the lab between hospital appointments, corresponding with colleagues until the end. She died in 1958 at 37, with over 40 published papers behind her. 4 years later, Watson, Crick, and Wilkins received the Nobel Prize. Franklin's name was not mentioned once. The full story only emerged decades later through historians who dug into notebooks and correspondence that had never been examined. Watson's 1968 memoir made things worse. He portrayed her as cold and difficult, a caricature that people who knew her said was unrecognizable. What the scholarship eventually confirmed is that Franklin didn't just contribute to the discovery, she made it possible. And even if she had lived, it remains genuinely uncertain whether the scientific culture of that era would have given her the credit she was owed. Today, a Mars rover carries her name. Her portrait hangs in the National Portrait Gallery. Students learn who she was. That's progress, but it's the kind of progress that arrives too late, in the way it always does for people who were wronged before the world was paying attention.